So Dr. Mark Belmonte is a professor of biological sciences at the University of Manitoba. He received his BSc and his MSc from the University of Calgary before moving to Winnipeg, where he did his PhD in plant science in 2008. So you are from here, really. After a brief postdoctoral fellowship at UC Davis, Mark moved back to Winnipeg to start his own lab in the Faculty of Science, where he's a full professor. Dr. Belmonte's group uses cutting-edge, next-generation molecular tools to improve crop production and protection in some of Canada's most important agricultural crops. He's published in over 60 times, been the recipient of numerous awards, including CBC's Top 40 Under 40 and the Canadian Society of Plant Biologists Research Excellence Award, and has been recognized for his outstanding contributions to research, teaching, and outreach by the Winnipeg Foundation and the University of Manitoba. He's devoted to promoting science education and research at outreach events across Canada and takes pride in training the next generation of young scientists. And with that, can we welcome Dr. Mark Belmonte, and he's going to talk to us about using RNA interference to protect crops against fungal pathogens. Thanks, Mark.
That's great. Thanks very much for that kind introduction, and also for the invitation to be able to come out today and speak to everyone, and also for putting on a great couple of days of agronomy learning. So what I wanted to do today was talk a little bit about the research that we're doing back in my lab in biological sciences, and that's in the development of what we like to call next generation molecular fungicides. And more specifically, what we're interested in doing is developing the next generation of crop protection tools, specifically protecting one of Canada's most important crops, canola. So I don't really need to introduce canola. After all, it is one of Canada's most important crops. But globally speaking, it is one of the top 3 oil seeds that's grown around the world. And if we take a look at its production globally over the past decade, we see a 23% increase in total yield. But because canola is so popular, we also know that canola can succumb to a number of different types of biological stresses. And one of the most important biological stresses that canola comes into account for is the fungal pathogen Sclerotinia sclerotiorum. And a recent disease survey shows that over 80% of all of the canola crops grown around the world showed some signs of Sclerotinia disease. That means there is an immediate need to find new, innovative, as well as sustainable tools and technologies in order to protect one of the world's most important crops. So before we get into the technology itself, I think it's really important to understand the biology. And if we can understand the biology for this plant microbe interaction, then we can certainly better understand how to develop these types of new cool tools. So the first thing we have, if we look at the bottom part of this screen here, are these sclerotia. So these sclerotia are these tiny little melanized structures. And these tiny little melanized structures can last within the agroecological system for over a decade. So they're incredibly durable as well as resilient. When the conditions are right, they're going to germinate and they're going to release ascospores. And the ascospores are airborne. They're then going to land onto the surface of the petals. And then they're going to co-opt those petals in order to provide all of the really good, the goodies and the nutrition. And those spores are then going to germinate. And once they germinate, they're going to start to collect all of that nutrition, and it's then going to land onto the surface of the leaf. And then once the petals land onto the surface of the leaf, it's going to start to penetrate through the epidermis, down into the mesophyll, and eventually down into the vasculature. And once Sclerotinia gets down into the vasculature, it's game over, right? So that infection becomes systemic. Sclerotinia is then going to travel down the stem. It's going to create architectural weakening of the plant, and then the plant is going to rot, and as well it's going to lodge. So has a tremendous impact on one of canola's most important yield robbers. And then that cycle is going to continue. It's going to continue over and over again. We're going to see more sclerotia. And so, there's really a need to find these new types of technologies to be able to understand how to protect one of the most important crops. So not only do these sclerotia last within the soil for over 10 years, but there's really no true genetic resistance to combat Sclerotinia, unlike some of the other fungal pathogens like blackleg. So what is our solution? Well, we're using what's called RNAi, or RNA interference. And what RNA interference does is that it interferes with the processing of genetic information within the cell, specifically at the RNA level. So what we do is we synthesize what's called a double-stranded RNA. And this double-stranded RNA shows sequence homology to some sort of gene that's found within the fungal pathogen or within Sclerotinia. So what we'll do is we will synthesize that double-stranded RNA. That double-stranded RNA will then come into contact with the cell. It's then going to get taken up by the cell in a process called clathrin-mediated endocytosis. Once it enters the cytoplasm of the cell, it's then going to get recruited by dicer. And dicer is going to do exactly what it says. It's going to start to dice up these molecules. It's going to dice up these molecules into these short interfering RNAs. Once it cuts them up into these short interfering RNAs, it's then going to be able to target a specific mRNA that's found within the cell. And if that mRNA is essential for the pathogen, so if that mRNA is essential for growth, if that mRNA is essential for development, and if that mRNA is essential for pathogenesis, that when we get rid of it, when we interfere with it, when we knock it down and when we knock it out, the idea is that we can also slow down the growth of the fungus or ideally kill the fungus. So why are we interested in RNA interference technology? Well, there's a number of great advantages to RNAi. Number one is that this type of technology is highly specific. So what we can do is we can design these types of next generation molecular fungicides all the way down to the gene level, but not all the way down to the gene level, but also all the way down to the species level. So what this type of technology is, it's a species-specific molecular fungicide. And so the idea here is that we would start to move away from some of the more traditional broad-spectrum fungicides as well as pesticides, because we know that there certainly are beneficial fungi as well as beneficial insects that are out there. This technology is also considered to be environmentally friendly. We can target an individual species, we can target an individual gene without having any sort of unintended off-target effects. This type of technology is also highly adaptable, which means that we can go into our database, we can identify genes that we think are essential for pathogenesis or growth as well as development. We can then synthesize these molecules, and then we can test these molecules in a lab setting within a couple of weeks. This type of technology is also really flexible. So not only can we develop a traditional spray that would get put onto the crop, usually at flowering to control Sclerotinia, but we can also develop transgenic plants. That means that we can now get the plant to synthesize these double-stranded RNA molecules that specifically target a gene that's found within the fungal pathogen, thereby conferring durable resistance across the entire life cycle of the crop. So what exactly is our strategy to protect canola using RNA interference technology? Well, the first thing is that we have to somehow access an RNA database. And so what we did is that we did mRNA sequencing of Sclerotinia isolates, not only Sclerotinia that was grown on plate, so in vitro, but also Sclerotinia that was grown on tolerant lines of canola as well as susceptible lines of canola. So we generate huge big data sets. We then made an algorithm in order to identify what we think are the best leads or the best targets, called a target identification pipeline, that uses our knowledge of the way that these genes are expressed, the way that these genes accumulate, the way that these genes are regulated. Once we've identified a set of targets, we then make sure that there's no off-target effects. So we have access to a tremendous amount of genomics literature as well as databases that's publicly available, so that we can, at least to the best of our knowledge, ensure that the molecules that we're designing have no off-target effects. So again, that these molecules are highly specific to an individual species and more specifically to an individual gene. We can then go ahead and synthesize these molecules in the lab. We would then spray these molecules onto the fungus and make sure that the gene that we're trying to target is in fact knocked down in the presence of RNAi. And then once we see and confirm that this gene is knocked down, we can then get into the cool functional analysis. So we can go ahead and develop that spray where we can spray onto the leaf and then infect the leaf with Sclerotinia and look to see what that disease looks like. And then if those leads and those genes start to look good, then we can also get into a transgenic approach. So how do we go about infecting canola for these large-scale screens? Well, we use a petal inoculation assay in the lab, and so the petal inoculation assay is a very similar type of assay that we would see out in the field. So for example, what we would do is we would take canola petals, and more specifically, canola petals that are senescing, so they're dying off. We would then inoculate those petals with ascospores. We then allow those petals to germinate, and then we'd inoculate the leaf. So we'd spray the leaf with our double-stranded RNA. We'd then apply the petal onto the surface of the leaf, and then we would look to see how disease was taking off within a matter of days. So what this graph is showing is a number of our targets. So far we've looked at over 100 different Sclerotinia genes, and we've characterized 100 different Sclerotinia genes within the lab. This graph is showing about 65 of those. And what each one of those bars represents is the amount of disease that we would see on a canola leaf. And so what we can see from this graph is that the vast majority of the targets that we identified actually reduced the amount of disease that was found on the leaf. More specifically, we're applying only about 200 nanograms of double-stranded RNA onto each leaf, and so we're screening through literally thousands of these things just to make sure that what we're seeing is real. And overall, we're seeing a reduction anywhere from 15 all the way through to 85%. But then we also have some of those anomalies where sometimes we actually get the promotion of disease. And so what we're thinking is what's going on with these ones is that these are negative regulators of pathogenesis, which means that if you were to knock down something that's negatively regulating, you're actually going to promote disease within the plant. So what exactly does this type of protection look like? And so here we have just some of, again, our favorite targets. So the first one on the top left-hand side there is a plant that was just inoculated with Sclerotinia. So this one didn't have any double-stranded RNA. We can also use other double-stranded RNA controls that are on the leaf. So for example, something that wouldn't necessarily target the gene of interest or a gene within Sclerotinia itself. And we see very similar results. So Sclerotinia really is a bully. It's going to infect canola very, very rapidly. And so what we're looking at here is disease within a matter of days. And so this is a necrotrophic pathogen. So again, really difficult to control. So, we get this really, gnarly looking lesion relatively quickly. And then just looking at a couple of our favorite leads here. So SS2 is a target that's looking at toxin biosynthesis. SS6, looking at protein synthesis. Again, these are all targets within Sclerotinia. And then also SS11, which is targeting a gene that's responsible for mitochondrial activity. So what we're seeing again is a nice reduction in disease. So we're not necessarily killing the fungus itself, but we're certainly slowing down the ability of Sclerotinia to infect the plant. So one of the really cool things about this type of technology is that it's easily adaptable, right? So in this case, earlier we're saying, okay, we can control Sclerotinia, or at least we can try to manage Sclerotinia using RNAi. But we also want to try to control or manage other types of diseases. And some of the other types of diseases that we're interested in exploring was botrytis. And so botrytis is a closely related necrotrophic pathogen to Sclerotinia, also infects hundreds of different plant species, has also called gray mold. And what we can see here from this graph is that we're looking at similar homologs in botrytis to our favorite Sclerotinia genes. We would then infect canola with botrytis, and then we would look to see whether the amount of infection was actually reduced. And what we can see is in all of the 5 leads that we explored here, we saw a dramatic as well as profound reduction in overall disease severity, not only in Sclerotinia, but also now in botrytis, which again makes this type of technology highly applicable. So we published this a few years ago with a bunch of my grad students as well as undergrads in the lab. So if you are interested in these types of methods as well as the developing of this type of technology, this is all available freely online. So one of the other things that we were interested in is how exactly are these types of RNA molecules or these double-stranded RNAs getting into the cell. So what's really cool about this stuff is that we can label these double-stranded RNAs, and we can label these double-stranded RNAs with the fluorescent marker, so in this case with fluorescein. And so what we did is that we inoculated Sclerotinia with these fluorescently labeled double-stranded RNAs, or these fluorescently labeled molecular fungicides. And then we used different types of microscopy tools in order to study how these molecules were getting taken up to the cell in real time. And so what we have here are a number of fungal hyphae at 2 hours, 4 hours, 6 hours, and 8 hours. And what we can see is that these fluorescently labeled molecules were specifically targeting the advancing tips of the fungal hyphae cells. and that this was occurring within a matter of hours. So we started to dissect this pathway even further at the molecular level, and so we started to use different types of inhibitors, we started to use different types of genetics, we started to use different types of RNAi in order to interfere with a process called clathrin-mediated endocytosis. And clathrin-mediated endocytosis is a way that different types of eukaryotic organisms can take things in and process that cargo within the cell. And so what's going to happen is that we would take our double-stranded RNA, that double-stranded RNA would then come into contact with the cell. And then it would get taken up by the cell in this structure, and this structure is that clathrin vesicle. And then that clathrin vesicle has a number of different components that are found on it, and then it's going to be able to target it to different regions within the cell. So we started to dissect this at a number of different levels, but we had a number of outstanding questions. So for example, is there some sort of double-stranded RNA receptor that's found on the fungal cell, or are these types of molecules just getting taken up by absorption? We also don't know fully just yet how exactly these double-stranded RNAs are processed within the individual subcellular compartments of the cell, but we did have enough information to conclude that clathrin-mediated endocytosis was in fact involved in the uptake of these molecules. So if we can better understand the uptake of these molecules, we can also predict how resistance might develop within the fungus and develop different types of ways to overcome that resistance that will inevitably occur over time. So we talked a little bit about the development of what's called these sprays, right? So these next generation molecular fungicides. But we also realized, we can get the plant to do all of the work for us. So instead of relying on the timing of the spray, so again, similar types of timing between 20 and 50% flowering, now what we can do is we can start to engineer the plant. We can start to engineer the plant to synthesize as well as make these double-stranded RNAs for us. And if the plant can make these double-stranded RNAs within every single cell of the plant, that means in theory, we should be able to see protection across the entire life cycle of the crop. So what we did is we took our 5 favorite leads or our 5 favorite genes. We made 35 different independent insertion lines. We took these plants all the way up to the T3 generation to make sure that what we were seeing was durable over time. We screened through thousands of different plants and thousands of different lesions that were measured to really prove that this technology is viable at the transgenic level. So what we're looking at here is just a couple of different examples. One within Westar, so that's the universally susceptible line, so it's universally susceptible against the vast majority of plant pathogens, as well as one of our favorite lines that we're abbreviating here, SS1. We did a number of different types of inoculation assays, so one just doing a leaf inoculation assay, and then the other one is a stem inoculation assay, which is more of an industry standard. And then we went ahead and quantified the amount of disease that we were seeing within not only the leaf, but also within the stem. And what we can see here is a dramatic reduction in the amount of the lesion that's formed within the leaf. Again, it's not completely resistant per se, so we do see some of the fungus that's starting to infect the plant, but it certainly is slowing it down. And when we take a look at the stems, the stem that, again, the universally susceptible line that was inoculated, we can see all of that necrotic tissue that's starting to form within the stem, and then that lesion is quite a bit larger compared to the transgenic line. So we went ahead and quantified a number of different components of the transgenic. So the first one is looking at the size of the lesion, the next one is looking at the amount of fungus that was found within the plant, and then the other one just making sure that the gene that we were targeting was in fact knocked down. So we take a look at the lesion size, and this was a lesion size that was found within the leaf. We saw a 45% reduction in the size of the lesion. We saw an 85% reduction in the amount of sclerotinia fungus that was found within the plant. Again, we use quantitative PCR in order to validate this. And then again, using qPCR, just making sure that the gene that we were targeting was in fact knocked down. So what we're seeing is that durable expression of these double-stranded RNAs certainly does improve plant health. So we like big data in the lab, so we did a bunch of next-generation RNA sequencing just to make sure that what we were looking at was real. We did small RNA sequencing just to make sure that the small RNAs that were generated as a result of these constructs were being processed in the right way. And then we went back to some tools that I started to pick up all the way back in my master's degree in Calgary, and that was using microscopy in order to really visualize what might be happening within the plant. And so we took a couple of different approaches, one using scanning electron microscopy, and then the other one just using really old school hand sections to be able to better understand the relationship between Sclerotinia as well as canola in either the universally susceptible lines, so the untransformed lines, or what we call the host-induced gene silencing lines, or the transgenic lines. So if we take a look at the untransformed lines, what we can see here is all of those fungal hyphae are starting to colonize the central component of the stem. And again, once it's colonizing that central component of the stem, it's systemic, and that plant is going to lodge as well as fall over. But in the Higgs line, one of the really cool things that we started to see was this band of electron-dense signal right here. And so this band of electron-dense signal suggested that there might be some sort of physical barrier that's preventing the fungus from moving from the outside of the stem through to the inside. Again, this was all using the mycelia plug assay on the stem. So again, using our RNA sequencing data set, we were able to see that, well, suberin was coming up. So suberin was coming up across its entire biosynthetic pathway. We then went ahead and used stain that was specific for suberin. And what we noticed within the transgenic line is that that same electron-dense, what we like to call force field, was actually full of suberin. So again, providing some sort of waxy physical barrier, preventing that fungus from penetrating deep into the stem. Again, just providing an additional level of resistance, and more specifically, physical resistance to prevent colonization of the plant. We then did whole plant assays just to confirm, again, what we're looking at was real. And so with our susceptible line is pretty much being wiped out once we sprayed the plants with ascospores. However, in our transgenic line, we can see that it's still producing. We got some really nice flowers, and then we went ahead and quantified and just made sure that we were getting some seed back. And again, we were getting some, again, really nice seed in each one of our independently transgenic lines. And one of the other things we were interested in is whether we saw a reduction in the sclerotia that were also produced. So again, Sclerotinia is going to produce these sclerotia within the stem, and they're going to, again, overwinter, so that's going to continue that cycle year after year, again, which makes it very difficult to control. So not only do we see an increase in seed production, but we also saw a reduction in the amount of sclerotia that were being produced within the plant. So what I wanted to do today was just provide a little bit of information about some of the cool stuff that we're doing in the lab, where we're driving innovation through discovery science. We know that Sclerotinia infects hundreds of different plants globally and causes billions of dollars in damage to growers that affects their bottom line every year. We've now developed what's called a topical next-generation molecular fungicide that's able to protect the plant against disease, but also took it one step further in the development of transgenic plants that show durability against Sclerotinia across their entire life cycle. So the idea is that we can now use RNAi technologies as a solution to improve global food security. So this work was carried out by a number of folks in the lab. So this was a picture taken a couple years ago, again, just to highlight and prove that every once in a while we do go outside, and every once in a while we do go out into the field, and we dose up on Claritin and make sure that we can survive the 5 minutes out there. I know you can tell I'm a lab guy, right? It was a success. And of course, all of their funding would not have been possible without the generous support of a number of funders. Thank you very much.
Thank you, Mark. That was a great presentation. I'm sure we'll have some questions on this. Can we start with Brianne right here?
I am going to ask about weeds. Thanks, Mark, for the presentation. So RNAi has been looked at for management of herbicide-resistant weeds to knock down some of the resistance mechanisms. But it kind of went quiet in the industry at the formulation of the spray component. So I was wondering if you could talk about some of the challenges to formulating those topical sprays.
So most important is how do you get it into the plant? And so with crops like canola, which have a really waxy cuticle, it makes it exceptionally difficult to get these types of products into the plant so that they can become systemic. What's great about this type of technology for fungal control is that you're really just trying to control what's ever going on the surface of the plant, so you don't need to worry about what's going to go on inside. And so the biggest challenge is how do you keep the spray active on the surface of the plant? So we've done experiments to show that the molecules are stable on the surface of the plant for a matter of at least 2 weeks, which is great. But the challenge certainly is getting it inside. And so there's different types of ways that you can get it. You can use diatomaceous earth. You could use different types of adjuvants, surfactants to try and penetrate. And then also, how do you keep those molecules on the surface of the leaf, and how do you make them bioavailable over a long period of time? And so there's different types of nanotechnologies that are currently being used. And so Need Emitter in Australia is using this bioclay. And so these are nano sheets of clay where you can essentially bind your dsRNA molecules to it, and then that would help as a sticker. But it's a challenge because plants are smarter than we think.
I think Tammy's got a question.
It kind of builds on that, I guess, or takes it from the other side. So with some of the targeted spray applications of the double-stranded RNA, are there any chemical or physical properties of the leaves themselves that'll help or hurt efficacy, stability, in controlling those pathogens?
Yeah, so the waxy surface of the cuticle automatically acts as just another level of structural resistance. So, in oil seeds like canola, you have that wonderful protection. But other leaves that might not have either hairs, or they might not have, so soybeans got a lot of hair, but other plants that might be have a little more of a flimsy leaf, you'd definitely be able to get those molecules in, but they're also naturally going to be more susceptible to pests and pathogens.
I have a question about how will this fit into the regulatory framework of pesticides and herbicides? Will it be able to be developed faster, slower? What are your thoughts on that?
Yes, so we're currently working collaboratively with different regulatory agencies in order to move this technology ahead, because they see a lot of value in this, and so they're aware of this type of technology, and that they're aware that this could be something that would just add another tool for the grower's toolbox in terms of crop protection, crop production. And so they're aware of it, and they're very supportive of it, is the short answer of the answer here.
Yeah. Okay, thank you very much. My question is, don't you see the Sclerotinia evolving over time?
Yeah, and so that's a really great question. And so that's actually one of the new projects that we're working on in the lab is looking at a directed evolution study or experimental evolution study where we'll force Sclerotinia to become resistant to these dsRNAs. So, and then look to see how that might happen. And so one of the ways that we're getting around that is that so far we've looked at a structure called the long linear double-stranded RNA, but now what we're doing is we're changing that structural conformation. So we're changing the structure of the RNAs to now what we call paperclip RNAs. So instead of having two open ends of the double-stranded RNA molecule, now we're capping those molecules. And so when you cap the molecules, it makes it less amenable to some of the other enzymes that would naturally degrade those types of molecules that are found within the cell, so nucleases as an example. So we're changing the structure of the molecule, and then also looking at different types of tools to get around that possibility of resistance. And there was a study done, so collaborator Steve Wired in biological sciences, we're doing a parallel study to control flea beetles using the same type of technology, so I know everybody's excited about that one. And so with flea beetles, there are artificial lab populations of flea beetles that are resistant to double-stranded RNAs. And so there's definitely a need to be able to understand how that resistance mechanism occurs and find different ways to get around it.
Do you guys have another one, or can I go again?
Go.
Okay, so I might murder this, but let's try. So a couple of questions. Do you think and I think you already sort of touched on this, do you think it is possible to achieve true qualitative resistance using endogenous RNAi, or is some level of infection needed in order for the pathogen to uptake the siRNAs? And the second part is, very interesting work on identifying homology to target both Sclerotinia and botrytis. Just like a fungicide, how broad of a pathogen profile do you think it would be possible to target using RNAi?
Yeah, okay, so I remember the second part of the question, so we'll go with that one first. So in terms of how broad can you get it, you can get it as broad as you have homology to the different genes. And so in this case, as long as you have 21 nucleotides that's similar between the gene sequence that you're trying to target, you should in theory be able to knock every single one of those down. So that means that you can make this as specific or as broad as you like it. And so one of the other things that we're actively investigating in the lab is this idea of off-target effects. So how many sequences do you have to have that are off or mutated that will have some sort of off-target, so that's some sort of homology. And then, would you mind
Okay,
repeating
on the first part, I
part?
think where we're going is, you get true qualitative resistance using that endogenous RNAi, or do you need some level of infection before that starts to be expressed, basically?
I think it's going to be really difficult to achieve true resistance within this system, but the idea, I think, is that just slowing it down has remarkable benefits. And so even if you can just slow that pathogen down, you're going to have a huge, as we've seen, just a huge promotion in terms of the way that the plant's going to respond, and will still generate seed for you. And so I think it's going to be difficult to achieve true resistance. And then I think it's like how the molecule stays onto the surface. So really at this point, the fungus just needs to be able to absorb the double-stranded RNA. So as long as it's found on the surface of anything, it'll be able to take it up.
Well, thank you, Mark. That was a great presentation. We really appreciate you coming, and we have a small gift for you here. Thanks so much.
Thank you.